Good morning and welcome all to our third Shanks Pathology Ground Rounds lecture today. It gives me great pleasure to introduce Dr. Samra Turajlic, who is, excuse me, who is a medical oncologist who's based at the Royal Marsden. And Samra has a stellar career. She did her undergraduate medical studies at Oxford and her clinical training at UCL. She did a specialist training in medical oncology before she was appointed as a consultant at the Royal Marsden in 2015. Prior to that, in 2013, she did a PhD in melanoma genetics and became a clinician scientist fellow dealing with or addressing predominantly cancer evolution. And in 2019, she became an independent group leader at the CRUK, the Crick Institute. So she has a number of roles externally also with ESMO, and she's the leader of the GCIP melanoma from the 100,000 Genomes Project. And she's been given a number of awards, including the ESMO Translational Researcher Award in 2022. So without further ado, it gives me great pleasure to introduce Samra, and she'll be speaking about tumour evolution and revolution and its transition to and crosspoint to pathology. Thank you very much, Samra.
Thanks very much for that kind introduction, and thank you, Sarah, for giving me the opportunity to speak to all of you today. A lot of what we do in the lab has involved pathologists over the years. That's always been a very enlightening interaction for me, so I'm really delighted to share some of our work with you. The title that was advertised was a little bit more exciting, talking about tumor revolution. But I'm hoping to excite you about tumor evolution instead at the intersection of pathology. So as Sarah says, I'm a clinician scientist. So the questions that we pose in the lab are very much informed by observations in the clinic. And one of the greatest challenges in oncology is really understanding the course of individual cancers and divergence in disease trajectories between patients with a same cancer type that might appear to have the same phenotype through routine clinical methods. And as a result of this, of course, we all appreciate that there is a proportion of patients that will be overtreated, where we misclassify their risk at the baseline. And of course, there is convergence in other patients onto disease recurrence, metastasis, and treatment resistance. Now, our group has taken an evolutionary view of this process to see if we can refine understanding of these disease trajectories at the beginning and inform both prognostication and prediction of therapy responses. So the way that we approach this is very much multidisciplinary. It starts with patient cohorts, and that's because the temporal and spatial scales of clonal evolution are very difficult to recapitulate in preclinical models, given that tumors evolve over decades and often evolve to population sizes that go well over 1 trillion cells. At the same time, what we're looking at when we deconstruct tumor evolution in patients' tumors is sort of the end of that trajectory and sort of inferring what happened before. And essentially, that's not a repeatable experiment, at least not in the context of sporadic cancer, where it's usually just a single instance of cancer in a single individual. So it's very important to complement these data with preclinical modeling. And here we deploy two methods. One are sort of mathematical simulations. These are informed by the parameters that we observe in patients. And here we can essentially rewind tumor evolution to get at sort of interactions that might be hidden in the patient data. And at the same time, we are working with patient-derived models. So these are tumor-derived organoids, iPSC-derived organoids, which we couple with genome engineering to understand the dependencies between the events that we uncover in patients in the course of tumor evolution. And then lastly, and this is certainly where pathology very much comes into play, we're interested in measuring these evolutionary features at scale. Certainly the approaches that we use in the clinic in our lab at the moment are too labor intensive and too complex to be scaled up for routine clinic use. And of course, we do want our work to have a clinical impact. So this is an important part of our lab program. So just to give you a quick overview of what I'll be talking about today, I'll be focusing on kidney cancer in the interest of time. We do work on melanoma as well, but perhaps I could leave that for another time. In the first part of the talk, I'll talk about evolution of primary tumors, followed by metastasis, followed by immune changes under therapy. And then lastly, I'll talk about that fourth quadrant, which is capturing evolutionary features at scale. So you'll all be familiar with clear cell renal cell carcinoma, and I hope to convince you that this is really the most useful and elegant model of clonal evolution, the understanding of which can be very helpful for application in other cancer types. So the cell of origin is posited to be the proximal convoluted tubule of the nephron. And we established a few years ago that the very first event, which intriguingly occurs decades before the actual malignant transformation, is an aneuploid event. So this is loss of the short arm of chromosome 3. This occurs in normal cells, and for reasons that we're investigating at the moment, these normal cells tolerate this aneuploid event, and it's probably fixed in a few hundred cells, and therefore the risk of kidney cancer is essentially the likelihood of picking up a second hit, which would lead to malignant transformation. The other intriguing thing is that this event usually happens through chromotripsis, which is a catastrophic genomic rearrangement that results in a shattering of chromosomes. And in this case, the result of that is an unbalanced translocation between 3p and 5q, such as this 3p is lost and 5q is gained. Now, the reason why we put so much emphasis on this first event is firstly that it's obligatory. It's present in almost all clear cell RCCs. But second is really the genetic composition. If we zoom in here of 3p, it contains 4 well-described tumor suppressor genes, VHL, PBRM1, SETD2, and BAP1. And so even if you didn't know anything about ccRCC genetics, you could very easily predict from here that the further clonal evolution will proceed through picking off the remaining alleles of these tumor suppressor genes. So just with that initial hit, essentially the cells are achieving loss of heterozygosity in 4 tumor suppressor genes. So it's a very powerful constraint on what happens subsequently. And indeed, the most common second event is a mutation or methylation in VHL. And this is essentially what leads to malignant transformation, that first hit. VHL does have a pleiotropic effect. I've included a few references here relative to many functions downstream of VHL. But of course, the one that manifests very strongly in ccRCC is the pseudohypoxic response. So essentially under normoxic conditions, VHL protein acts to target HIF1 alpha and HIF2 alpha for proteasomal degradation, stopping them from upregulating transcriptional pathways that lead to angiogenesis. On the other hand, if VHL is genetically deleted, this regulation no longer exists, and therefore, even in the presence of normal oxygen tension, there's upregulation of HIF pathway and then downstream transcription. And of course, this is what we've taken advantage of, this pseudo-hypoxic state in terms of therapy developments in ccRCC, and for best part of last 20 years, we've been targeting this pathway mostly with VEGF receptor TKIs, but more recently there's been approval of a direct inhibitor of HIF2 alpha, which is belzutifan. So that's been a very exciting development. So in spite of reaching this cancer hallmark of angiogenesis, biallelic inactivation of VHL actually is not a strong driver of tumor growth. And majority of the tumors that we examine will have mutations in one or more of these remaining tumor suppressor genes. So the conundrum here is that we have these very narrow, very stereotyped genetics in this disease, but extremely varied clinical behavior. And if there are any oncologists online, you'll appreciate that there is a really bewildering range of clinical behavior that we see in the clinic. Increasingly, these tumors are detected incidentally. These are small renal masses. We know from observational studies that a great majority of small renal masses never progress into more advanced tumors. On the other hand, we have more locally advanced tumors that might relapse with some latency and might relapse with this kind of intermediate metastatic efficiency, presenting with a solitary metastasis or oligometastasis. This is relevant clinically because these patients can be managed with surgery, even in the context of stage 4 disease. And then lastly, we have patients who might be treated with curative intent relative to their primary tumor, but will rapidly develop comprehensive metastases around the body, suggesting that the presence of occult metastasis from the start. So we really set out to try and explain this clinical diversity in the context of those very narrow genetics by applying an evolutionary framework. And essentially what we find is that those 4 genes that I was mentioning have very highly conserved patterns of order and combination, so mutual exclusivity, co-occurrence, and conserved ordering, such that, let's say, genotype A only ever occurs on the background of genotype B, or that genotype A and genotype B are mutually exclusive. And what this results in are essentially different modes of evolution. So you see something that we refer to as linear evolution in this first setting. There is very little clonal diversification. We're really looking at a very monoclonal tumor here, where either through lack of mutational supply or lack of selective pressure, there's not an awful lot of evolution happening. Initially, when we discovered this evolutionary subtype, we thought that this was perhaps a lead time bias. We were catching these tumors at too early a stage of evolution to really detect the evolutionary potential. But when we calculate the time from the most recent common ancestor to diagnosis, these tumors are frequently in place for a median of 30 years. So there is really something that's sort of constraining further evolution in these tumors. Now, these tumors in the middle evolve in a very typical Darwinian fashion. So it's sort of incremental changes over time, resulting in highly heterogeneous tumors where there is fixation of, in parallel of multiple subclones that are genetically distinct. And these subclones are likely to have similar fitness, which is why they're able to fix, and there is no sort of overall winner, if you like. And these tumors also grow over long periods of time, but sort of pick up their growth towards this sort of evolutionary, more advanced end of the trajectory, because the clones that are being selected are increasingly more fit, but they do not cause a clonal sweep. And contrast that with this last group, which we refer to as punctuated evolution. So in this setting, these are also relatively homogeneous monoclonal tumors, but what makes them different to this group early on is that they pick up a highly fit clone. There's sort of accelerated evolution, and that highly fit clone, which usually is characterized by multiple driver mutations and fixation of multiple aneuploidies, drives a complete clonal sweep out, competing everything else. And these tumors grow to a very similar size to these tumors in the middle, but they do so over 4 years. So in the literature, this has been described in the context of other cancer types, including uveal melanoma and triple-negative breast cancer. These tumors really represent a great challenge when we think about early detection, because that window would be very narrow, given to rapid growth and as you will see in a moment, rapid metastases from these tumors. So when we consider what selection is acting on, of course, it's not the genetic alterations, it's the phenotype that's afforded by those genetic alterations. So we began next to think about this interaction between the genotype and the tumor microenvironment and what kind of phenotype that might give us in the context of these different modes of clonal evolution. And we hypothesized that in the context of branched evolution, where you're needing to support many different subclones in parallel, that this might reflect what's in ecology referred to as a cooperation phenotype. So here there would need to be promotion of local tumor infrastructure to support all these different subclones. And we posited that punctuated evolution reflects a colonization phenotype where there is rapid turnover of cells and there is quick selection for motility because the environment is too harsh to promote these rapidly proliferating cells. But phenotypes are very difficult to measure in situ. I mean, certainly we often use bulk transcriptome, for example, which we can deconvolve into, to some degree, to tumor signals and tumor microenvironment signals. But of course, as pathologists, you will know that tumor histology not only offers information on phenotypes, but these are also spatially resolved. So one of the things that we struggle with when we're studying clonal evolution is that sort of sampling bias and spatial bias, where there is lots of physical segregation of these clones in the tumor, and that's exactly where pathology can help us. And so this is work that has been led by an incredibly talented pathologist in the lab who just recently completed her PhD, Charlotte Spencer. And she posited that the tumor microenvironment changes could be reflected in tumor architecture, summarizing here the type of architecture that's observed in CCRCC, and that cell intrinsic features would be reflected in tumor cell morphology. And again, summarizing here these sort of conserved features that you will all know well. Finally, because vascular angiogenesis is such a prominent feature of TME, we were very keen to extract vascular features at scale. And Charlotte worked with mathematicians as well as AI team here at the CRIG to develop this high-performing vascular feature extraction algorithm. And she did this initially by doing a large-scale staining, IHC staining with CD34. She was then able to extract vessel masks and then apply these simply to an H&E to extract these components of the vasculature. And then at the same time, as I said, she was working with a mathematician to extract the most important features in this feature map of vasculature, and then through dimensionality reduction, arriving at a set of features that can be extracted from the H&Es. And when we map these features, vascular features, back to these modes of evolution, what we see is that they separate out very clearly. So in these evolutionary subtypes, where there is very slow evolution, very heterogeneous tumors, we see that there is a continuous vascular pattern, as you can appreciate from here, which is more dense. This is the U map for vascular features. You can see that they're mostly on this left-hand side of the U map. While in these tumors that are growing through punctuated evolution, this sort of supercharged evolution, if you like, the vasculature is more discontinuous, the pattern is more sparse, and again, as you can see, there is a fairly good separation of vascular features in the UMAP here. Now, what about tumor cell features? Well, Charlotte thought that the best way to extract these would be to develop a pipeline, a CNN, to try and predict through machine learning algorithm the genotypes that are sort of canonically associated with these different evolutionary trajectories from the H&E slides, and then through an explainable AI approach, understand the features, the salient features that the machine learning algorithm is using in order to predict those genotypes that underlie different evolutionary trajectories. And just to recap, PBRM1 is associated with that Darwinian evolution, while BAP1 is associated with more punctuated evolutionary patterns. So now when we map this back onto our evolutionary trajectories, Charlotte observed that the cell morphology in tumors with branched evolution was very typical of CCRCC with clear cytoplasm and round nuclear, while cell morphology in these opposing tumors was very distinct with eosinophilic cytoplasm and irregular nuclear. And of course, this would be reflective of high grade and mitochondrial density, which would go perfectly with what we know phenotypically characterizes these tumors. So essentially, to conclude this first part, what we have done is to build an understanding of these modes of tumor evolution, which sit in a particular context of tumor vasculature and cell morphology, which associate with different modes of tumor progression. And then lastly, what I haven't shown you this, it's published work, we can also predict the benefit from especially surgical intervention in the context of stage 3 or stage 4 disease by considering these evolutionary features. So this has really convinced us that continuing on this pathway of understanding tumor evolution could have important clinical impact in due course. So moving on to metastases now, I've already shown you that we are able to, and this is through multi-regional molecular profiling, to resolve these phylogenies in tumors and understand how these clones fix over time and their relationship to each other. Now, when we have a primary tumor like that, and then we also have a metastatic tumor that's either occurred synchronously or metachronously, we have been able to trace back the origin of the metastasis to a clone within the primary tumor. And this was important because in the metastasis research community, there is a lot of debate around where metastases emerge. So there are two opposing views, sort of linear evolution of metastasis, where metastases are believed to evolve at the site of the primary tumor, or parallel evolution of metastasis, where maybe one of these earlier clones leaves the primary tumor site, sits in another site, and then evolves in parallel to the primary tumor. In the context of CCRCC, each time we examined the metastasis, we were able to trace back its identical genetic clone within the primary tumor site. And I think this has important implications also when we think about the benefit of surgery, especially in the context of cytoreductive nephrectomy, perhaps reducing this sort of burden of metastasis-competent clones that could feed additional metastases. Now, we're able to ask a very simple question when we do this, because we can ask what the features are that distinguish these clones that are able to make a metastasis versus those other clones that have failed to make a metastasis. And we see that they have a very similar burden of mutational events. So there is no difference in the profile of those genetic drivers that I've been mentioning. But there is a profound difference when it comes to larger scale alterations, so chromosomal copy number alterations across the genome. And obviously, these are the types of events that are affecting expression of hundreds, if not thousands, of genes at the same time. And in many ways, this makes sense if we think about the many phenotypes that are required for the metastatic process. So we see elevated levels of WGII. Now, this is an index that simply reflects the proportion of the genome altered by copy number. So if you like, it's sort of tumor aneuploidy burden. And in particular, we see bottlenecking for these two specific events. So loss at chromosome 9p and loss of 14q are very highly enriched within these metastasizing clones. Now, this perhaps didn't come as a surprise because 9p loss certainly had been linked to poor prognosis in CCRCC in the past. But what we were able to resolve here is that the way that it does that is through driving acquisition of metastatic competence. So again, coming back to this concept of, what's selection acting on? Well, it's not acting on 9p and 14q per se. It's acting on whatever phenotype these karyotypic changes are inducing. And so we repeated this exercise of following clonal evolution, but now at the level of transcriptional changes. And just giving you an example here, we might have a tumor that initially was wild type at 9p, and then through tumor evolution had acquired loss of 9p. And very simply, we asked what are the transcriptional differences between these earlier 9p wild type clones to 9p lost clones. And what's really important here is that you're controlling for any patient factors that might influence transcriptional changes. So that's where this type of cohort really offers a special opportunity to understand the specific changes related to acquisition of a genotype or a karyotype. And essentially what we see is quite a lot of transcriptional changes that happen upon 9p loss and 14q loss. And the most pronounced is overexpression of proliferation pathways. So this is telling us that metastatic competence, at least in part, is reflecting that proliferative capacity. Because 9p loss and 14q loss often co-occur, it's quite difficult to deconvolve in these bulk transcriptome data which one of them is driving this phenotypic potential. And so we resorted to reanalyzing single-cell RNA sequencing that's publicly available from almost 1 million cells of ccRCC. And we can use this algorithm called infer-CNV, where we can subset those subclones that have 9p loss versus subclones that have 14q loss. And essentially what we see is that it's 9p loss that really drives, in these volcano plots here, drives the proliferation pathways, so overexpression of E2F targets and G2M checkpoint, while we see absence of that in 14q. Furthermore, we suspected that immune evasion might be an important component of the metastatic phenotype. And when we, again, this is at the level of individual patients, when we relate what happens in the wild type clone as it progresses to the 9p lost clone, we see clear depletion of CD8 T cells, while this is not the case with 14q. So this would suggest that it's 9p loss drives immune evasion while 14q doesn't. This is not to say that 14q is not playing a role. And right now we're using genome engineering methods to understand what the likely cooperation is between these two events. But what might be happening relative to 9p is that this is essentially acting in cis, because if we look at the region on 9p, of course, CDKN2A and B are involved, but there is also this interferon cluster. So plausibly, if you deleted both of those, you could increase proliferation, but also enact immune evasion. And indeed, a mouse model of PDAC, where both of these clusters were deleted, this is work from Scott Lowe at Memorial Sloan Kettering, was shown to drive metastasis as well as immune evasion. Now, more recently, we've been looking at the exact breakpoints of the 9p lost region. And of course, this is only one allele. When we look at the second allele, we sometimes see inactivation of the remaining alleles of CDKN2A or B, but we don't see encompassment of this interferon cluster. So we decided, because of that, this sort of inability to pinpoint what's driving 9p loss, to model it. And this is a method that we've just recently developed. And this is the ability to generate aneuploidies right across the genome for individual chromosomes. And this has allowed us to have paired lines. So this is the parental line. This is chromosome 9. You can see that this is copy number neutral. And now when we apply this method, which we call CRISPR-T, we generate a line that has loss of, this method normally generates whole chromosome losses. So this is a whole chromosome loss at 9. So what we recapitulate when we now RNA sequence these clones is upregulation of E2F targets. However, we don't see any obvious downregulation of interferon, as was seen in that PDAC model. So that suggests that additional factors beyond aneuploidy and beyond proliferation are required for metastasis to be formed, and perhaps these are further phenotypic features or might be environmental features. So this sort of makes us reflect on our earlier work. So one of the things we're able to do with the type of tumor sampling that we do, and through this sort of clonal resolution of individual biopsies, is for every clone, we can provide an exact coordinate within the primary tumor. And then when we do that systematically, we can ask questions about how clones expand and grow. And so in relation to these aneuploid clones and metastasizing clones, we were able to ask across many patients, well, where are they found in the tumor and how do they grow? And essentially what we see that is that these clones that are highly aneuploid drive very large clonal expansions, and that goes well with what we've seen about that upregulation of proliferation. But spatially, they're more likely to be located in the center of the tumor. And furthermore, we find that this area of the tumor where metastasizing clones emerge often associates with large areas of necrosis. And of course, as pathologists, you'll know far better than I that necrosis is an important prognostic marker in CCRCC, but we now provide sort of a biological rationale for that. And of course, what might be happening here, there's many potential scenarios because we don't know the direction of the interactions. And I've just popped down these factors here. So we know that aneuploidy is important. So aneuploidy emerges through chromosomal instability, which is variation, which is chromosomal missegregation. We know that when aneuploidy is fixed in a cell and it propagates, that this gives those clones heightened fitness. And we know that one of the features is increased proliferation, so there's increased cell turnover. Now, if that increased cell turnover is such that its needs cannot be met by the vascular supply, you will get a true environmental hypoxia, so not the genetic pseudohypoxia that's caused by VHL, but also accompanying features such as low pH. This, we posit, leads to necrosis. Now, necrosis is a poorly understood mode of cell death, but we know that relative to apoptosis, it's a much more pro-inflammatory way of cells dying. And then whatever conditions that creates, it leads to selection of more aneuploid clones. Those non-aneuploid clones cannot survive in this type of environment. And so we posit that this sort of set up a vicious cycle of ongoing selection for metastasis. But as I said, we don't really know the directionality here. So we've started picking these factors off sort of one by one. And the first thing that we've started looking at is how these clones that have high levels of chromosomal instability evade the immune system, because we know that in the context of chromosomal instability, the presence of cytosolic DNA in the cytoplasm will trigger, or should trigger in healthy cells, cGAS-STING signaling. So what happens downstream of activation of this signaling is upregulation of type 1 interferon response, which should have tumor suppressive effects. And at the same time, cGAMP is a key signaling molecule that can also license antigen-presenting cells, such as dendritic cells, to activate their own internal cGAS-STING signaling, and again, type 1 interferon response, which will lead to immune stimulation and should lead to anti-tumor killing. And of course, this has been the basis of the therapeutic development of STING agonists. So we have this paradox, really, because as clonal evolution proceeds, as I've shown you, we get increasing levels of chromosomal instability and aneuploidy. But then when we look at T effector cells, these earlier clones are more immune infiltrated than these later clones that have become aneuploid. So how do they avoid cGAS-STING signaling? And so we've again returned to this idea of looking at this question within individual patients as they go from regions of low aneuploidy. So this is low WGII, it's that index of aneuploidy burden, to regions with high aneuploidy. These are the main chemokines that we predict are upregulated downstream of intact cGAS-STING signaling. So CXCL9, 10, and 11, and we see in regions of low aneuploidy they're present, but in regions of high aneuploidy they're absent. So this would suggest that the regions with high aneuploidy are avoiding this cGAS-STING activation in some way. And what we noted is that in these regions of high aneuploidy there is upregulation of these potential STING repressors, SLC19A1 and EMPP1. Now, how might this work to bypass cGAS-STING activation? Well, SLC19A1 is a transporter of cGAMP. And if you remember, cGAMP is the key immunostimulatory molecule that licenses immune cells to activate an interferon response. And we posit that when this is overexpressed by the tumor cell, it leads to reabsorption of cGAMP, and therefore there's less cGAMP available to generate anti-tumor immunity. ENPP1 is a degrader of cGAMP, and not only does it therefore reduce the amount of cGAMP available, but degradation of cGAMP leads to accumulation of adenosine, which in itself is immunosuppressive. And this has been shown very nicely in the context of breast cancer by Samra and MSK, who's really is very much a key leader in this field of cGAS-STING signaling to lead to immune suppression and even cause immunotherapy resistance. So now we go back to our sort of original work where we showed that high levels of aneuploidy correlate with poorer survival in patients with kidney cancer. But we're now able to provide some more nuance in this prediction. So when we take into account the status of cGAS-STING signaling, so this is high aneuploidy, but intact cGAS-STING signaling, these patients do well in spite of having high aneuploidy. But when high aneuploidy is combined with high levels of cGAS-STING suppression, you can see that these patients do badly. So I hope I've shown you in this second part that cell intrinsic features of metastases are aneuploidy, increased proliferation, but these are not sufficient and require cell extrinsic features need to find themselves in a particular tumor microenvironment in order to metastasize. So I will maybe I might skip the part on immunotherapy if that's okay, because I would be keen to share with you something that's very close to pathology. So I hope I've convinced you that these types of conclusions and insights are really very reliant on having that sort of spatially resolved data. And the sort of elephant in the room relative to both research setting and clinical setting is that the proportion of the total tumor that we sample for molecular profiling, again, in both settings, is really tiny. Now, if we were dealing with a hematological malignancy, that would be fine because you have a clonally mixed population. Every time you take a blood draw, you're getting a representative sample. But as I've shown you, and as is appreciated across all solid tumors, there is a lot of physical separation of subclones, and there's very little mixing. So what that means is that not only is there undersampling, but there is unrepresentative sampling because something that might look like a major clonal expansion in one region of the tumor might be completely absent from somewhere else. And as I mentioned already, this sort of multi-regional sampling that we do for research purposes is rather labor intensive. And maybe just to illustrate this a little bit further, even when so single region biopsy might have 500 million cells in it. When we do these multi-regional biopsies, we're of course spatially differentiating. We are capturing more of that spatial heterogeneity, but we're still capturing a relatively limited number of cells when a representative sample might run into trillions of cells. And so I've already mentioned these figures. So these were gotten through sort of clinical audit of how much material goes up for molecular profiling, and this is based on TCGA. And this sort of undersampling scales up with tumor stage. Of course, as you get larger tumors, the undersampling is more relevant. And again, considering this emergence of intratumor heterogeneity and evolving tumor and more advanced tumor, we've really become very vulnerable to that sampling bias. So a number of years ago, we were trying to think how to overcome this issue. And normally when I speak about this topic, this sort of fact about incineration, about leftover fixed tissue, is usually met with some surprise, but of course it won't be a surprise to you that after you've completed your sampling for pathology examination, any leftover tumor after a period of time is incinerated. So what we started doing is sort of taking over this leftover fixed tissue and then homogenizing it, essentially to get over that issue of lack of clonal mixing and to force these clones to mix. And therefore, then when you, at least in theory, you have identical sample aliquots, and when you try to reconstruct your tumor clonal solution, you would get a better resolution because you have representation of more clones. And arguably, if you think about biomarker data, you should get a better resolution of those as well. So in the initial pilot of this, we saw that representative sequencing on the back of this tumor homogenization really overcame intratumor heterogeneity. So we got a much more reproducible performance, as you can see up here, rather than using individual biopsies where you might be hitting a different clone each time. We discover more variants, as you would expect. We get a more accurate clonal structure by only profiling this one sample, and we get a better inference of the variant allele frequency across the tumor as a whole. And then more recently, we've added another step to this, which is fixed, which is flow sorting of these fixed cells. So initially, we were motivated to do this in order to reduce redundant sequencing and really enrich the homogenate for tumor cells as opposed to stromal cells. And in some instances, we're able to get the tumor purity as close to 100%. And then, of course, when we sequence, you will detect more variants, and you also need to do less sequencing. So we thought, well, this will be economical, but it also helped us to increase clone detection. But the other opportunity that's emerged from this is actually to really sort for stromal cell phenotypes as well as tumor cell phenotypes. And this is the idea. Of course, we know that the TME and biomarkers such as PD-L1, for example, are also heterogeneously present across the tumor. And now if we homogenize the sample and flow sort for different stromal cells by their markers, we're able to get a sort of representative picture of the tumor microenvironment. Of course, this loses the spatial resolution, and the bit of my talk that I skipped will have highlighted the great relevance of spatial resolution, and you'll appreciate that more than I. But what we're getting at here is the possibility of really getting a representative readout of different cell abundance, but also the abundance of different states. So, for example, you could ask, well, how many exhausted PD-1 positive T cells are there in this tumor, etc. And we have been able to do this in the context of a prospective trial now, which we have been running at the Marsden. At the moment, we've got to about 300 representative tumor samples collected. This is a breast cancer cohort. I'm just showing you this briefly. So this work has been led by another very talented pathologist who I've had the honor of working with in the lab, Brian Hanley, who's coming to the end of his PhD. And what Brian has been able to show so far is that the leftover tumor after diagnostic sampling typically contains most of the cancer cells in the resection specimen. So this is all the blocks, and this is the representative sample. So we really are picking up a representative sample here through homogenization. And second, the great concern was about formalin artifacts. So Brian has spent a lot of time developing this bespoke bioinformatics pipeline. And in the end, we can see that when we filter the variants that we get involved for these formalin artifacts, we get a very similar burden of variants as TCGA, which is fresh sampling. And of course, the quality of these samples is better than FFP sequencing because you don't have the addition of paraffinization, everything that goes with that. So first of all, we looked at the molecular landscape, and we see that it looks very sensible. Representative sequencing involved recapitulates this existing driver landscape. Now, these are clonal mutations, and as you would expect from representative sequencing, because we're capturing more of the tumor in a mixed fashion, we're picking up more of these subclonal mutations. And what's more, we're picking up these genes that are exclusively subclonally altered. And of course, it would make sense that these genes don't necessarily come up in single sample studies, which are really heavily weighted. So our sort of knowledge and understanding of driver genes is heavily weighted for clonal genes because that's what you usually pick up in single sample analysis. In the interest of time, I'll just go quickly here. We see that this now recapitulates the landscape, but also we're identifying both clonal and subclonal biomarkers. And actually, there is a large number of subclonal actionable biomarkers that we're seeing. I do want to show you this last part because, again, I think it'd be great to get your feedback on it. So Brian has done a large-scale exercise looking at intra-tumor heterogeneity of Ki-67. And while in some tumors, it's fairly stable across different FFP blocks, in other tumors, it really varies hugely when he scores it across different blocks. So then we took this average of Ki-67 by immunohistochemistry and then compared it to what we get as Ki-67 through flow sorting of the representative sample. And essentially, the representative sample gives you that modal population. And when it comes to decision making, of course, Ki-67 is a prominent feature of some of the prognostic panels that are used in breast cancer. We can see that depending on which block has been used, this might alter the outcome. And what we're really excited about is that we are now able to do some of this phenotype-based phylogenetic reconstruction. So, for example, here we're just molecularly profiling those proliferating subclones that are Ki-67 positive. And we're able, so here in red, we can see that some mutations are exclusively present in these proliferating clones. And this is, for those of us studying clonal evolution, it's a very exciting perspective. So the conclusions from this part, and of course you will appreciate this, that biomarkers are susceptible to sampling bias. But I hope I've shown you as well that this method of homogenization, representative sequencing, can overcome some of these issues. And I've shown you a couple of recent improvements that build on this method. So it remains me to thank the wonderful people in the lab who do this work. I've acknowledged many of them as we've gone along. Also, it's impossible to do this type of work with patient cohorts without the clinical research infrastructure at my unit at the Marsden. So I'm extremely grateful to everyone there, many of our collaborators, funders, but most of all, patients and their families who really underpin this research. And just to say for any early career researchers online that we have both doctoral and postdoctoral opportunities in our lab, specifically for pathologists. Now, I could stop sharing unless it's helpful to have the slides up to refer back to them. What would you suggest, Sarah?
Well, thank you very much, first of all, Samra. I think perhaps if you don't mind, if you could stop sharing, and then we can take some questions. So I can't see any at the moment in the Q&A, so I will start off with some myself. So first of all, perhaps working backwards, I mean, just fantastic overview of all of the work that you're doing. It's just really sensational. But first of all, with respect to the FACS sorting of the tumour, the blended tumour samples, so are they fixed or are they fresh? Is that fresh material?
They're fixed. So we essentially take what's left over after pathology reporting sign off. So that's all fixed. It's then macro dissected to separate out tumor and normal. We're also looking at tumor margin. And then these three types of samples are homogenized separately. This provides then kind of an infinite analyte, if you like, for all the downstream applications. So if we think about all those instances where you're kind of running out of FFP material, to especially in the context of oncology, where you might be prioritizing patients for 3, 4, 5 trials down the line, that feels important. And obviously, it's a well-mixed sample for the flow sorting. And of course, we've had a lot of skepticism about flow sorting of fixed cells. So we've done a lot of benchmarking against FFP, IHC, and so on to show that these are representative populations. So following homogenization, there is a dissociation step of these, and then we've essentially optimized something like 30 antibodies so far. So initially we started off with just epithelial cell markers because we're mostly working with epithelial tumors. We were doing some sorting by aneuploidy on the premise that tumor cells were more likely to be aneuploid and sort them that way. But as I've shown you at the end, we can really, I mean, any antibody that you can optimize for FACS, we can use to separate out populations of interest.
It's fantastic. And sorry, I know there are a couple of questions that have come in the meantime, but I'm just sort of wondering how long does that take, and how have you gone back and compared the results to the diagnostic biopsies or diagnostic resections that were then issued as reports?
Yeah, yes, we have. So Brian has done a lot of comparisons to the molecular profile from single FFP blocks. And essentially what you get from those is a very long tail of variants that are only present in that block, which goes with the kind of subclonality and illusion of clonality. And very clearly, because we're looking at sort of multiple blocks within the representative sample, we are getting better estimates of what the true sort of cancer cell fraction is, and we're able to separate out those minor subclones, which if you think about drug targeting, if your drug target is only present in 10% of the cells, then your therapeutic window is going to be very narrow.
Yeah, okay. And that's great. I think it would be brilliant if we could try and expand this out into other tumors, obviously. So there's the first question from Dr. Farahat is that, do you think it's still a long time for treatment options that only targets the metastatic clones?
Yeah, no, that's a great question. And actually, I think we're guilty in the oncology and therapeutic development community of sort of ignoring aneuploidy drivers, somatic copy number alterations. It is a lot of focus on gene function and targeting individual genes and genetic mutations. But increasingly, and this is not just in kidney cancer, it's appreciated that metastases are driven by these large-scale alterations. So I think the approach can be twofold. I mean, one is sort of targeting genes that promote chromosomal instability. So we know in kidney cancer, for example, BAP1 is very relevant for promoting chromosomal instability, as is SETD2. Those are tumor suppressor genes. They're not oncogenes, so they are a bit difficult to target, but easier than just targeting aneuploidies as a whole. And the other is kind of finding these interactions. As I've shown you, aneuploidy is not sufficient to drive metastasis. It really needs to interact with a particular tumor microenvironment. So, if we can reduce hypoxia, if we can reduce necrosis, if we can re-establish cGUS signaling, all those things would kind of counteract the phenotypic consequences of aneuploidy.
Great. Great, thank you. And then the second question from Ian Tomlinson. What happens if an early renal cell cancer has a copy number loss of heterozygosity at VHL? Does that tend to guide it down a route that doesn't involve the other 3P genes?
That's a beautiful question. Thank you, Ian. And it's very timely because we've just completed an analysis of a large-scale analysis over 1,000 tumors on the background of VHL disease. So here there is a germline VHL mutation, and then the canonical pathway would be losing 3P. But in a small number of lesions, actually, we are finding either a mutation or methylation of the second copy of VHL. So now you've not lost 3P, and that's sometimes constrained by the fact that it's not only VHL that's deleted in the germline, but there is a neighboring gene called BRIC1, which is an essential gene. So basically, you can't lose 3P. It's kind of an evolutionary dead end. So indeed, if you don't lose the whole of 3P, we don't see mutations in any of those additional tumor suppressor genes. And that actually then impacts the phenotype. These tumors stay very small. They have no metastatic potential. So that's a great question. Thank you very much. I hope I answered it.
Okay, and I think we've got time for one last question. That's from Marnix Janssen, who says, fabulous talk. Common clinical conundrum is that working up patients is, of course, detecting small kidney lesions. Is it aggressive and detected early or indolent? How close are we using evolutionary maps to answer this question?
Yeah, thanks, Marnix. Great question. Obviously, small renal masses are an issue. We're currently profiling, we're focusing on small renal masses exactly for the reasons that you outline. And actually, we're not seeing any exclusivity relative to these genetic trajectories. I mean, of course, you get more clonal evolution with greater population size. So I think the answer is going to be multiparametric. I don't think the genetics are going to be sufficient. I think we're going to have to get some tumor microenvironmental signaling, combine those two together. And then I could answer Benjamin's question just very quickly. Yes, we are really astonished that we find these clones in large areas of necrosis. It's like an extinction event. But I can tell you from our observation of trying to derive organoid lines that they come from these areas of necrosis. The most aggressive clones that we're able to establish in the lab, they're born in these areas of tumor necrosis.
That's great. Well, thank you very much, Samra. Really fantastic tour de force lecture. I've certainly got more questions and I might ask them by email. But I can just follow up on Samra's last slide, saying that if there are opportunities to work in her laboratory, it would be an excellent environment for any PhD student. So please take her up and on that potential offer. So with that, I thank you very much, Samra, for your lecture today, and thanks all for attending this Grand Rounds lecture series, which is sponsored by the Jean Shanks Foundation, together with the Pathological Society. And we're just going to advertise the next lecture, which will be mid next month. Thank you very much, Samra.
Thank you so much, everyone.
Have a good day.
Good to see you. Bye-bye.